Welcome back to The Explainer. Today, we're diving straight into a fascinating, highly rigorous scientific process, how environmental scientists actually track and measure invisible carbon monoxide emissions using a standard known as EPA Method 10B. If you've ever looked up at a massive industrial smokestack and wondered how we actually know exactly what's coming out of it, well, you are in the right place. To really understand this, we're going to follow the exact chronological journey of a single gas sample. We'll start with what Method 10B is, then move to catching the gas sample, lab analysis using GC and FID, quality control and calibration, and finally, calculating those hard final numbers. All right, kicking things off with Section 1, what exactly is Method 10B? Simply put, EPA Method 10B is the Environmental Protection Agency's precise regulatory playbook for extracting and quantifying carbon monoxide at industrial sites. We're talking specifically about petroleum refineries and other designated stationary sources. So we're not talking about the exhaust coming from your car's tailpipe here. This is about massive, complex industrial operations where measuring an invisible, odorless gas is absolutely critical for both environmental compliance and public safety. Moving right along to Section 2. Catching the gas sample. Out in the field, this is where the journey physically begins. Now, the big catch here is that we can't just measure raw exhaust gas directly. Why? Because industrial emissions are basically a messy soup of different compounds. Things like carbon dioxide and various organics act as imposters, and they'll completely interfere with our lab analysis later on. So most of that interfering CO2 has to be stripped away first. We do this by passing the raw sample through an alkaline permanganate conditioning system. It effectively scrubs out the unwanted gases, leaving us with a much cleaner, conditioned sample to evaluate. So the physical journey right at the source breaks down like this. Step 1, we extract the integrated gas sample. Step 2, we scrub out those interferences through that conditioning system we just mentioned. And step 3, we safely collect the purified sample in what's known as a Tedlar bag, or an approved equivalent. This bag is super important because it acts like a secure, non-reactive vault to safely transport our sample from a messy industrial site back to a pristine laboratory setting. So, the big question, how do we actually measure what we literally can't see? Well, before we haul this sealed sample bag into the lab to uncover the final data, please take a quick second to hit that like button on this explainer and subscribe to the channel. It really helps us out, and it ensures you never miss out on our deep dives into the science of environmental compliance. All right, let's head into the lab. Section 3, Lab Analysis with the GC and FID. Let's see what happens when the heavy machinery gets involved. Once we're in the lab, we don't just crack the bag open and take a sniff, obviously. We inject the gas into a highly rigorous analytical setup. First up, it hits a gas chromatographer, or GC, which uses a separation column to filter out any last residual CO2 or organics that might have survived the field scrubbing. Next, it passes through a reduction catalyst, and finally, it reaches the Flame Ionization Detector, the FID. This thing is incredibly sensitive, capable of reading carbon monoxide in a minimal range of just 10 to 1,000 parts per million. And of course, everything is logged by a permanent data recording system. Now, here's where the chemistry gets really cool. You might be wondering, why do we even need that reduction catalyst? Well, it's because the Flame Ionization Detector is specifically designed to measure hydrocarbons, like methane. It actually cannot see carbon monoxide directly. No way, right? So our reduction catalyst performs this brilliant little chemical magic trick. It converts the isolated carbon monoxide into methane, CH4. By throwing this chemical disguise onto our sample, the FID can now accurately detect and quantify it. It's just a fantastic scientific workaround. Okay, moving to Section 4, Quality Control and Calibration. Because honestly, all this complex machinery doesn't mean a thing if the equipment is off. To keep the system honest, we run some vital checks. For instance, before we even touch the real sample, we run a carrier gas blank check. This ensures that any accidental contamination in our baseline gases is adding less than 5 parts per million of positive bias. We also run a reduction catalyst efficiency check to make sure that chemical magic trick we just talked about is actually working perfectly, keeping any negative bias under 5%. And to top the whole thing off, we never just test a sample once. We run all samples in triplicate, that's 3 separate injections, to absolutely guarantee the precision of our analytical results. Finally, Section 5, Calculating the Final Numbers. Let's step away from the machines and look at the actual math. First things first, we have to account for water. If we see visible condensation inside our Tedlar bag, we use what's called equation 10B1. This calculates the moisture content fraction, which we call BW.
We figured this out by taking the vapor pressure of the water in the bag and dividing it by the barometric pressure in the analysis room. We need this specific moisture fraction to make sure water vapor isn't artificially inflating or deflating our final numbers.
Once we've got that moisture fraction, blow, we plug it right into the denominator of our ultimate goal, equation 10b-2. We combine it with A, representing our average sample area from those triplicate lab tests, and R, which is the mean calibration response factor of our machines. When we crunch this equation, it strips away all the variables and hands us the exact dry basis carbon monoxide concentration in the bag. Seeing all these intricate steps laid out really leaves us with a provocative thought, doesn't it? Without this incredibly methodical process, from the initial field scrubbing to the chemical transformations in the lab, right down to the strict equations that even factor in the barometric pressure of the room, how could we possibly hold stationary sources accountable for the air we all breathe? It is exactly this level of unseen, rigorous science that turns an invisible gas into a concrete, measurable reality. Thanks for joining me on this explainer, and keep questioning the science behind our world.